You know, it's like sometimes the biggest discoveries come from the most unexpected places.
Right.
Like you might be, going through a bunch of old files or something and boom, you stumble on like a huge historical revelation, or
Exactly.
maybe you're in the lab and you're doing just like a routine check and all of a sudden like something totally remarkable pops up.
Yeah.
And that's kind of what we're going to be diving into today.
Okay.
We're going to be looking at some recent evidence regarding the presence of residual DNA
Right.
in mRNA COVID-19 vaccines,
Okay.
specifically Pfizer and Moderna formulations. And chances are you've probably encountered at least some discussion around this already.
Yeah, absolutely. There's been quite a bit of information.
Yeah, there's been a lot of information circulating. And really what we want to do today is kind of focus in on what the recent findings actually indicate
Right.
and why they might matter for your understanding.
Exactly. And I think it's important to kind of, I guess, just recenter and focus on, like you said, what we're seeing in the findings.
Yeah.
And why this might be important to folks out there listening.
Precisely. And we're not going to be speculating or anything. We're basing this deep dive primarily on a recent article from the Pulse,
Okay.
which brings together findings from a number of independent laboratories,
Okay.
as well as insights from experts in the field.
Great.
So we're going to unpack these claims of DNA contamination, look at who made these discoveries, and perhaps most importantly,
Right.
what the potential implications could be.
Yeah.
So where did this kind of unexpected finding first come to light?
So the article highlights the initial work of Dr. Soňa Peková
Okay.
at Tilia Laboratories.
Okay.
She is a clinical biochemist and molecular geneticist with a pretty significant background in research.
Okay.
The article cites 71 published papers.
Wow. So
Yeah, and her analysis pointed to what she described as excessive amounts of residual DNA
okay.
in both the Pfizer and Moderna vaccines.
In both.
In both.
Wow. Okay, so now when we say excessive,
Right.
we're not just talking about like, oh, it's a little bit over.
Yeah.
We're talking about levels that were found to be up to 100 times higher
Wow.
than what regulatory guidelines permit.
100 times.
100 times. Think about it. Like if you had a speed limit
Right.
and someone was going 100 times faster,
Yeah.
like that's a massive difference.
It's not just like a little oopsie.
Yeah, right. Yeah, exactly. And what's even more surprising is that in some cases, the amount of this residual DNA actually surpassed the amount of the mRNA itself.
Right.
Which is the active component of the vaccine that's
Exactly, the part that's supposed to trigger the immune response.
Right, right.
So that's a pretty fundamental imbalance, isn't it?
It is, yeah. Like it really makes you think about the consistency of the product.
Of the product, yeah. And it raises a lot of questions about, the manufacturing process and quality control.
Right. How is this happening?
Exactly.
So Dr. Peková's findings weren't actually the first indication of this issue.
Right.
The article also refers back to earlier work done in April 2023 by Kevin McKernan.
Okay.
He's a genomics expert.
Right.
And he reported discovering DNA contamination in the bivalent booster shots from both Pfizer and Moderna.
So both of them again.
Both of them again, yeah.
Interesting.
And what was particularly notable about his findings was that he identified a gene sequence originating from the simian virus 40.
Oh, wow.
Or SV40.
SV40, okay.
Now, it is worth noting that his initial report was in the form of a preprint.
Right.
Meaning that it hadn't yet undergone formal peer review by a scientific journal.
So it's kind of like the first draft almost?
Exactly, yeah. So McKernan's work kind of flagged this earlier, and then Dr. Peková's findings seem to kind of reinforce those concerns with some quantitative data. But then we have this Professor Phillip Buckhaults from the University of Southern California.
Okay.
And his involvement is really interesting because he wasn't someone who initially believed these claims.
Right. He was actually, according to the article, he was initially quite skeptical.
Yeah, he was a skeptic.
Yeah.
So Professor Buckhaults is a very experienced molecular biologist and cancer geneticist with a very strong background in next-generation sequencing, which is a very sophisticated way of analyzing genetic material.
So he knows what he's doing.
He knows what he's doing, yeah. And initially, like I said, he considered these earlier claims to be a conspiracy theory.
Yeah.
He didn't buy it.
He wasn't convinced.
And he actually decided to conduct his own independent tests
Okay.
on samples of the COVID mRNA vaccines to see for himself.
Wow. So he really wanted to get to the bottom of it.
He wanted to get to the bottom of it. Talk about a turnaround, right? Like a leading expert in the field who initially dismissed the whole idea actually takes it upon himself to investigate.
That's pretty remarkable.
It is. So what did his independent analysis reveal?
Well, his findings were actually quite significant.
Okay.
Professor Buckhaults reported finding what he described as billions of tiny DNA fragments.
Billions with a B?
With a B.
Wow.
Specifically in Pfizer's mRNA vaccine.
Okay.
This led him to actually present his findings.
Oh, wow.
In a formal testimony at a South Carolina Senate hearing.
Okay.
Where he laid out the data from his lab.
So this is getting pretty serious now.
It's definitely gaining traction.
Yeah.
And it wasn't just a, technical presentation of data.
Right.
He also spoke with journalist Marianne DeMasi.
Okay.
And I think his reasoning for looking into this is actually quite compelling.
Okay.
He stated that he didn't start with any preconceived notion that there was something wrong with the vaccines.
Okay.
He himself is vaccinated, and he recommended it to his family.
Right.
However, he was also aware of reports of vaccine injuries, and he felt a responsibility to investigate potential underlying causes.
That doesn't sound like someone with an agenda.
Not at all.
Like pushing an agenda or something.
No, it sounds like a true scientist.
It does.
Just trying to follow the data.
Yeah.
And in that interview, he actually made a very direct statement.
Okay.
He said, quote, Kevin McKernan was right. This plasmid is present in the vaccine.
Wow.
That's a pretty strong confirmation coming from someone with his expertise who was initially a skeptic.
Right. And so what's really striking here is that these aren't just isolated incidents.
Right.
The article points out that more than 8 independent laboratories across the globe have now replicated this finding of DNA contamination in these vaccines.
So it's not just one lab or 2 labs. It's 8.
More than 8.
More than 8.
Wow. Reaching the same conclusions. Independently.
So that really suggests that this isn't just a one-off error.
Right.
Or, somebody messed up a test. Or,
right.
It seems like there's something bigger going on.
Yeah, this is like a systemic issue.
Potentially.
Yeah. Yeah, so the big question that naturally follows is, so what? Why should you, the listener, be concerned about this residual DNA?
Exactly. And this is where I think Professor Buckhaults' expertise in cancer genetics becomes particularly relevant.
Yeah.
Because his primary concern, stemming from his deep understanding of cancer genetics and somatic mutations, which are changes in DNA that occur after conception.
After conception.
Okay. But not the DNA you're born with.
Right.
But changes that happen
during your life.
Throughout your life.
Got it.
His concern revolves around the potential for this residual DNA to actually integrate into our cells' genomes.
Yeah.
Now, these DNA fragments are encased in lipid nanoparticles.
Okay.
You can think of these as tiny fatty bubbles.
Okay.
That are specifically designed to protect the fragile mRNA.
Okay.
And help it slip inside your cells. So they're like little delivery vehicles.
Yeah.
And Professor Buckhaults' worry is that these same lipid nanoparticles could also facilitate the entry of the DNA fragments into the cells.
Right.
Specifically into the nucleus, which is the cell's control center, where they might then become a permanent part of the cell's own genetic code.
So it's like inserting WHO code.
It's like splicing
into the software that runs the cell.
Exactly. And that's where the potential long-term concerns could arise.
That's the big worry, right?
That's the worry. Now, it's really crucial to emphasize that this is a question of potential risk.
Yeah.
It requires further investigation.
Of course.
To determine the actual likelihood and consequences of this happening.
We don't know for sure yet.
We don't know for sure. But Professor Buckhaults himself is quoted as saying, I think we should check and find out.
Right.
So he's not making definitive claims of harm.
Yeah.
But he's clearly highlighting a biologically plausible pathway that warrants serious attention and research to understand if it's actually happening and what the implications might be.
Absolutely. Okay, so the article also brings in the perspective of Professor Nikolai Petrovsky, who is an expert in immunology and infectious disease.
Right, an expert in the field.
What are his main concerns regarding these findings?
So Professor Petrofsky has also raised warnings about the high and inconsistent levels of DNA contamination being reported. His focus tends to be more on the quality control measures in place at the mRNA manufacturing facilities. He's basically questioning how these significant and variable levels of residual DNA could be present in the final product.
Like, how did this get through?
Right. It suggests potential shortcomings in the purification processes.
So it's not just the presence of the DNA itself. It's the fact that the amounts seem to vary quite a bit between different batches, which would point to inconsistencies in how the vaccines are being made.
Exactly. How is this getting through the quality checks? And Professor Petrofsky really stresses the urgency of addressing this issue. He cautions that if these elevated levels of DNA contamination are proven to have negative health consequences, regulatory bodies could face serious accusations of negligence for not having detected and addressed the problem earlier.
So this could be a really big deal.
It could be. He goes so far as to suggest that Robert Kennedy Jr., in his capacity as Director of Health and Human Services, should initiate an immediate and independent expert inquiry into this matter.
So that's a pretty strong call to action.
Yeah, he's not messing around.
No, he's not messing around.
So
the article also touches on the regulatory landscape surrounding this. And it seems like the existing rules might not have anticipated this particular situation.
That's a really key point. The article highlights that the current regulatory limits for what's considered a safe amount of residual DNA in vaccines were established before the development and widespread use of lipid nanoparticle technology.
So before this new delivery system was even a thing.
And this is crucial because, as we've been discussing, lipid nanoparticles are specifically engineered to enhance the delivery of genetic material into cells.
So the safety guidelines we're currently relying on were designed for a different type of vaccine delivery.
It's like having safety standards for traditional mail delivery and then applying them to drone delivery. The method of transport has fundamentally changed. The potential risks and how we need to assess them.
It's a whole different ballgame.
It is.
And so the concern is that these lipid nanoparticles might be making it easier for even small amounts of residual DNA to get inside cells compared to older vaccine technologies.
So the old safety thresholds might not be adequate for this new method.
So this definitely provides a lot to consider.
A lot to think about.
And you know, it's important for you, our listener, to remember that this deep dive is a focused look at the specific scientific findings and the expert opinions surrounding them. We're not making broad statements about all vaccines in general. We're concentrating on this particular issue of residual DNA in these specific mRNA COVID-19 vaccines from Pfizer and Moderna.
That's a vital distinction.
And as we navigate this information, particularly given how polarized discussions around vaccines often are. It's essential to keep in mind that our aim here is to impartially present the information as it's laid out in the source material.
Just the facts.
Just the facts.
No opinions.
We're conveying the discoveries and concerns raised by these experts without endorsing any specific conclusions beyond what is presented in the article itself. Our goal is to understand the evidence and the questions that it raises, not to take sides in any ongoing debate.
We're not here to tell you what to think.
Not at all.
We're here to give you the information.
Exactly. Okay, so let's quickly recap the key points we've covered.
Okay, so we started by discussing the initial findings by Dr. Peková, indicating significantly elevated levels of residual DNA in both Pfizer and Moderna vaccines. Then we talked about Professor Buckhaults' independent confirmation of substantial amounts of DNA fragments in the Pfizer vaccine, corroborating those earlier concerns raised by Kevin McKernan, including that presence of SV40 sequences.
And importantly, these findings have now been independently verified by more than 8 laboratories worldwide.
More than 8.
So it's not just a fluke.
It's a pattern.
It's a pattern. And we also highlighted Professor Buckhaults' concerns about the potential for this residual DNA to actually integrate into our cells' DNA, given his expertise in cancer genetics.
A very real concern.
Yeah.
And Professor Petrovsky raised concerns about the consistency and quality control in the manufacturing processes and the potential for regulatory negligence if these issues are found to cause harm. And then, of course, there's that regulatory context. You know, the fact that the current safety limits for DNA contamination were established before the advent of lipid nanoparticle delivery systems, which may alter how we need to assess the potential risks associated with residual DNA.
Yeah, like, are the old standards still appropriate?
Are they good enough for this new technology?
So it seems like the repeated detection of this residual DNA and the accompanying expert concerns definitely warrant closer attention and further investigation.
This is something that we need to keep looking into.
Yeah, absolutely. And this leads us to a final thought for you to consider. Given that the current regulatory limits for DNA contamination in vaccines predate the widespread use of lipid nanoparticle delivery systems, what are the potential implications for how we evaluate the safety of new delivery mechanisms in pharmaceuticals as a whole? It prompts us to think about whether our regulatory frameworks are keeping pace.
That's the question.
With the rapid advancements in scientific and medical technologies,
are we doing enough?
Are we doing enough
to ensure safety?
And how do we ensure they do in the future?
That's the big question.
That's a big question.
Yeah, lots to think about.
Lots to think about.
Thanks for joining us.
Thank you.